So thank you very much. What I'd like to do today is talk about studies on yellow fever vaccine. Yellow fever vaccine's been my sort of hobby for the last 25 years, and it's always interested me how such a great live attenuated vaccine works. We don't really understand much about the mechanism of attenuation, but hopefully I'm going to tell you a bit about it today, about at least why it probably works and how that can be used for quality control of vaccines. And I'm going to talk about a technique called deep sequencing. So we'll talk about yellow fever vaccine first of all. It's one of the most efficacious vaccines. Okay. I failed the first test. Ah, okay. Yeah. Yellow fever vaccine is one of the most efficacious vaccines in use today. It's a live attenuated vaccine that was developed in the late 1930s and has been used in about the last 75 years. There are only 6 producers of the vaccine worldwide, and they produce about 200 million doses of vaccine annually. The vaccine is produced in embryonated chicken eggs using technology that hasn't changed since the 1940s, and the reason for that is every time they try to transfer the technology to cell culture, it's never worked for one reason or another. Either the virus has changed or it hasn't grown well enough in cell culture, and so it's still grown embryonated chicken eggs using technology. It hasn't changed since 1945 with a seed lot system. In the last 75 years, some 600 million people have been immunized, and one dose of vaccine, which contains 10 to the 4 plaque-forming units, will give you immunity for at least 10 years and probably lifelong. So it's very attenuated and highly immunogenic. This is how the virus vaccine was derived from Mr. Asibi. Mr. Asibi was a gentleman in 1927 who had a mild case of yellow fever. He was bled. The virus from him was passaged from monkey to monkey, and that gave rise to the first wild-type strain of yellow fever from Asibi. And then that was taken and passaged, as I'll show you in the next slide, through chicken tissue to develop the 17D vaccine that we know today. So wild-type Asibi was taken and passaged through 3 different stages, 18 times through minced mouse embryo, 53 times through minced whole chick embryo, and then finally for 128 times in minced chick embryo without the nervous tissue, so they can get rid of residual neurological properties. This was 176 passes, and this gave rise to the so-called 17D vaccine. There were a number of other series of vaccines made at the same time, 17A, B, C, D, E, F, etc., but only 17D was attenuated. It was unique. Max Tyler tried to repeat the studies but never could, but he still got a Nobel Prize for his studies in 1951. So to summarize, a CB virus wild-type strain was passaged 176 times through mouse and chicken tissue. This gave rise to the 17D attenuated variant, and we now use substrains today, such as 17D204 as a substrain we use to vaccine today. Unfortunately, the 17D real attenuated variant got lost, so we can't really study it, so we only have the vaccines available today. Remarkably, the vaccine strain 17D has 3 great biological properties. Wild-type yellow fever causes liver disease, viscotropism, and the 17D has lost viscotropism. Wild-type yellow fever is transmitted by mosquitoes, and this virus is not vector competent, and also it's lost neurotropic characteristics. So in many ways, it's an ideal live attenuated vaccine. If we look at the genome of yellow fever virus, it's a single-stranded pulse-sense RNA virus molecule, 10,872 nucleotides in length, has a short 5' untranslated region, 3 structural proteins shown in red, capsid membrane envelope, 7 non-structural proteins called NS1 through NS5, and then a 3' untranslated region. Now, although we don't have the original 17D available, we do have the current vaccine strains, 17D204 series and the 17DD, and I'll talk more about this as we go along. But by doing deduction and detective work, you can compare the sequences of wild-type Asibi to 17D, different variants. You can identify there are 20 amino acid differences between the vaccine and the wild-type parent. 1 in the membrane protein and 8 in the envelope protein. So this is the structural changes. The pink ones here, there are a total of 9. And if we look at the non-structural genes, we find there are 11 amino acid substitutions, 1 in NS1, 1 in 2B, 1 in 3, 1 in 4A, 1 in 4B, 2 in NS5, and 4 in NS2A, plus 4 silent changes in 3' untranslated region. Now, we know from a number of studies that attenuation is multigenic, but we don't know actually that the mechanism of attenuation or what controls it, although we have to put different substitutions together to get an attenuated phenotype. And this has been a very hard study to do using infectious clone technology. But it really hasn't really helped us to understand how the virus is attenuated. It's the mechanism of attenuation. Now, if we look at wild-type RNA viruses, they are what we call quasi-species. That means to say they exist as a population of different RNA molecules with different sequences. The reason for this is positive RNA viruses like yellow fever have an RNA-dependent RNA polymerase, not a DNA polymerase. There's no proofreading, and in fact, it's highly error-prone. And this introduces mutations, so continually, it's like a swarm of RNAs of different sequence. And if you think of a yellow fever virus, the virus replicates once every 12 hours. You then have selection pressures through different hosts or cell types or tissue tropism. You've got a continual change in the virus RNA population. And if we look at the mutation frequency of wild-type West African yellow fever, we know it's somewhere around about 1 to 4 times 10 to the minus 4 substitutions per site per year. Now, if you think thank you very much. Okay, great, thank you. And if you think about the genome is 10,000 nucleotides, it means every time the virus replicates, it's going through at least one mutation. So it's continually evolving, and it has a swarm of RNA species. So we were interested in understanding what happened if you looked at yellow fever vaccine. Was it stable or did it mutate? And so our first study was we immunized 6 volunteers with the US vaccine YFVax from Sanofi Pasteur, and we bled them on day 6 when they were viremic, and we isolated the viruses. And it was really amazing the results we had, because we looked at the serum viruses, 1, 4, and 5, the entire genome. If you looked at the entire genome of serum virus 1, there was only 1 non-coding change, that's all there was. Looked at the virus 4, 1 change again, which was coding, the only coding change we saw. Serum virus 5 had 2 non-coding changes, again, nothing else is there. And then we looked at the structural NS5 for the other viruses, and really we only found 1 non-coding change. So really a highly stable virus, apparently, as determined by Sanger sequencing in terms of what happens to the vaccinee. And that may give us some idea of the attenuation is the virus doesn't mutate very highly, and that's why it's highly attenuated. So to get a better handle on this, we decided to move from Sanger sequencing, what's called the first generation sequencing, to get into deep sequencing technology. Now, to remind you, first generation sequencing is Sanger, a chain termination method. And this is looking at a dominant member of a population. So it's a consensus sequence, and we'll only see the dominant member. Whereas if we have a quasi-species, there's going to be plenty of mutations in the population. And one way of looking at this is to use second generation sequencing. It's termed deep sequencing, next generation sequencing, massive parallel sequencing to look at the RNA population. And so rather than looking at just the dominant nucleotide at a particular position, we can look at the whole depth of the population. And we've been looking at 50,000-fold reads. What that means is we can get down to a level of each nucleotide, look at it at 40,000 plus different members of the population to ask the question, is it diverse as a quasi-species, or is it uniform without any nucleotide variation? So we went back and took the Asibi virus and compared it to this batch of vaccine. And to give you an idea what we're looking at wild-type strain Asibi here, and we're looking at the YF vaccine from Sanofi Pasteur in the US, 237 passes later. Now, I'm going to talk about some of these other vaccines later on. This is the pedigree genealogy of yellow fever vaccine by different manufacturers, but let's look at the US manufacturer first of all. So we're looking at virus separated by 237 passes. This is the techniques to go through for deep sequencing. The Asibi virus we looked at was the lowest passage available. It's 6 passages in monkeys and 3 in mosquito cells. And the vaccine batch we used was just commercial vaccine that hadn't been passed, put in anybody, it was just normal vaccine. We go for a number of different techniques to actually sequence it, but it's sequenced on an Illumina HiSeq 1000 instruments. And we do a number of statistical analyses. And so when you do deep sequencing, it's all about statistics, because you're looking at such a large number of reads at 40,000 depth coverage. It's all about answer the question statistically, are there differences in populations? And given a 20-minute talk, I can't go into all this, so I'm going to hit the highlights. So the first highlight was when we looked at the consensus sequence, we wanted to ask the question, okay, if we deep sequenced it and we looked at the consensus, would it be the same as what happened with Sanger? And the answer was yes, and that was very reassuring. It told us that if we did Sanger sequencing compared to deep sequencing at a level of consensus, there was no difference. So we looked at Asibi and compared by Sanger sequencing to deep sequencing when we found 1 non-coding change at nucleotide 2704. When we took the YF vaccine, we found the sequences were identical. So it gave us confidence that when we looked at deep sequencing technology, it had representation of what would be seen in the consensus sequence. So I can show you data, but I'm going to try and summarize it to help you go through, because I know I have problems with some of the statistics. So when we looked at Asibi and 17D quasi-species populations, they intersect unidirectionally, and that means we find the vaccine variants are found in the Asibi population rather than the converse. And what this tells us is, overall, Asibi is a typical RNA virus with a quasi-species population, whereas the 17D vaccine is relatively homogeneous. It lacks what we call diversity. There's no variability in the RNA population. And this probably tells us what the mechanism of attenuation is. The virus doesn't mutate, it's highly stable, you can inject it into people, it doesn't mutate back, and that's why it's attenuated. Can we understand what that mechanism is? That's a slightly different question, and we'll come back to that as we go along. So what am I talking about diversity? What does this mean? So the simplest way of thinking about this is, if we had a coin, we can heads or tails, right? If it's heads every time, the probability of it being heads, there is no variability, so it's 0. If it's heads or tails, it's 0.5. It can be a head or it can be a tail. But when we start looking at nucleotides, it's a bit more tricky, because we can have A, C, U, or G. And so the total coverage, let's say, was 100 nucleotides. At that 100 reads, and we had 25% of each of the 4 nucleotides. If you do statistical analysis using diversity, also called entropy, we can use a statistical analysis called Shannon's entropy to understand the variability in the population. So Shannon's entropy says if every base was an A, probability would be 0 because it's no variability. If it's equal population, the maximum entropy you can get is 1.61. So let's go through and give an example. So this is an example where we have a coverage of 100 reads, 96 are A, 1 is a C, 1 is a U, 1 is a G. So the Shannon's entropy figure for this is 0.18, because it's not the maximum of 1.61 because it's not equal, it's mostly A, so the entropy is relatively low. So if we go and then look at Asibi versus 17D, and this is an entropy read going across the genome, as coloured across the genes, going across the whole genome, the 10,862 nucleotides. We're looking at Asibi versus 17D. The black dots refer to the nucleotide changes separated Asibi and 17D. And you can see the higher the Shannon entropy, the higher the number goes towards 1. And as you can see, there's far more variability in the population in Asibi than there is in 17D, suggesting, as I was saying earlier, that Asibi is a typical quasi-species population, whereas the population 17D is relatively homogeneous and doesn't have much variability, although it does at a couple of positions like there and in the 3' non-coding. So what does this mean? So if we looked, for example, at some sites to give you what we would see, if you looked at 1819, for example, you see here that 17D in 100% of the reads is a U, whereas if we look at a Asibi, it's a mixture of a U and a C. You can go through across here at different nucleotide positions, and you'll see with nearly every time, it's 100% of the nucleotides are one particular nucleotide rather than a mixed population. And the exception is here at 51, 53, where there's a small population of A. So that's what we see, relatively homogeneous population across the whole vaccine. So we're interested in taking this further and asking the question, what happens if you take vaccine from different manufacturers? And we have a collection of vaccine strains, the primary seeds, secondary seeds, and the vaccine from different manufacturers. There has been 12 manufacturers who have made yellow fever vaccine across the last 75 years. And if we look here at the slide, what we're seeing is Asibi to 17D. The 17DD substrain is only used in Brazil at the current time, but it's been used in Colombia and Senegal there as well. We have the 17D204 series of vaccines, which I talked about earlier, the YFVax in the US, but we have Senegal, France, Australia, USA, Netherlands, Colombia, England, India, and South Africa. And then more recently, the WHO derived a seed by the Robert Koch Institute in Germany, and the Russians, the Nigerians, and the Germans use this 17D 21377 seed virus. So, to cut it short, this is the viruses we use: South Africa, UK, US, Russian, Nigerian, and German, and that compared to Senegal and Colombia for 17DD. And here I want to make a point is we use YFVax, which is Sanofi Pasteur, but that originally, 20 years ago, was a national drug company before Sanofi Pasteur bought it. And that NDC means National Drug Company. And I haven't got time to go through it, but we can actually use deep sequencing to distinguish the manufacturer based on the heterogeneity in the population at particular positions, how it's changed between manufacturers. But when you do deep sequencing and you're trying to do phylogeny, it's not straightforward, and you have to do a technique called root mean square distance. And what happens here is, if we find overall figures, the Asibi is different to 17D, as we want to anticipate. The 17DD is different from 17D204, and the 17D204 and the 17D213 series can't be distinguished. They're genetically very homogeneous. So what do I mean by that? There's a horrible slide here, which I'm not sure you can read very well, but this is all the variability in the population between different vaccines we've analysed. The triangles represent non-coding changes. You'll see a few circles. They are coding changes. And then these bars here represent reversion back to Asibi, which is relatively rare. There's a fair amount of heterogeneity in the population. Now, I'm not sure you can read it well at the back, but a better way of looking at it is to look at a population as a histogram, where we're looking at entropy going this way. So the more the line goes this way, the more variability. And this is the amount of variability in a particular position. So as you can see, all the 17D vaccines really overlap each other. There really is very little variability, saying the seed-lot system is very good at controlling the vaccines. Now there's two exceptions to this, and this is shown down here in the population, where the Russian vaccine is more homogeneous than the other ones, and the one over here is in red here is the National Drug Company 6676 virus, which has more variability in the population than the other viruses. Now that's potentially very interesting because this is a batch of vaccine that failed the monkey neurovirulence test. It went into monkeys, 1 out of 10 monkeys died. The vaccine was not used because it failed the monkey neurovirulence test. If you sequence the virus by typical Sanger sequencing, you find that it's the normal sequence of a 17D virus. You can't distinguish it. Yet the variability in the population does show difference of heterogeneity. There is more variability in that virus as compared to the other 17Ds, suggesting that deep sequencing can be used as a technique to actually quality control vaccine batches of vaccine. So to finish off, to keep time, this is the first comparison of a live attenuated vaccine virus with its wild-type parent by deep sequencing. We found that 17D vaccine is less variable than the parental wild-type strain of Asibi by several independent measurements, and I haven't took time to go through all of those. It's clear that 17D vaccine was derived by mutation of a Asibi virus and not by selection of a virus in the Asibi population. The quasi-species population is fixed in the vaccine, whereas Asibi appears to be a typical RNA virus quasi-species population. And this probably explains the excellent safety record of the vaccine. There's limited drift between 17D204 and 17213 substrains, and you can't really resolve them, but it does show they have a seed-lot system for 17D that's highly homogeneous and gives the same vaccine time and time again. There is strong evidence for low diversity and sequence homogeneity among the entire 17D lineage. And I think this says that deep sequencing has applicability to be standardization and quality control of yellow fever vaccines in the future. To finish off, I'm a spokesperson for our group. Andrew Beck did most of the work, a graduate student in the lab, is very talented. A number of other people were involved, Tom Sheeb, Bob Tesh, Steve Wyden, Tom Wood, collaboration with Kate Ryman in Pittsburgh, and we had support from NIH and the Gilson Lombard Foundation. Thank you very much.
Thanks for the overview. So, any questions?
Yeah.
Excellent talk. I'm just wondering, why do you think there is a little variation in the yellow fever?
Why is little variation?
Yes.
It's because of the seed-lot system. When the vaccine was first produced in the late 1930s, many different laboratories got the vaccine and passaged it all sorts of times for use as vaccine. No one had really any ideas that passage history would make a difference. And what happened was it got passaged over 400 times, not the 176. And when it got to passage 400, the virus had changed. It became totally weakened, attenuated. It was over-attenuated, no longer immunogenic. And so in 1945, the WHO, was United Nations in those days, before WHO, they decided there needed to be a seed-lot system to control how the vaccine was made to stop this problem of over-attenuation. And so the idea was, you know, this genealogy I showed you where you have seed viruses, one pass to a secondary seed, and another pass to the vaccine that's used. So over the last 60, 70 years, we've followed that system where a manufacturer will grow a primary to make their secondary, then use a secondary to make the bulk, and then the bulk becomes the vaccine that's used later on. And then when you finish using your one bulk, you then go take the secondary again, regrow it to make more vaccine. and then every so often you have to go back to the primary. But the idea is a seed lot system allows you to make sure every vaccine is the same. And we find this is true by consensus sequencing or by deep sequencing, saying that the manufacturers using technology, it really works very well.
Any other question?
I'm glad that the yellow fever virus vaccine is highly stable.
Yes.
And when you look at the reading frame, I understand that this nucleotide sequence is highly conserved, but perhaps you can change the reading frames and then changing not just a single nucleotide, but maybe all 3 nucleotides so that they can be much more stable by using reverse genetics.
Yeah, so we've been playing doing that. It sounds as if it's very easy to do, but in fact it's not. And one of the problems is that doing reverse genetics is hard with yellow fever. The flavivirus plasmids are not very stable in bacteria. You have to use low copy number plasmids. It's very hard to grow them up. And then when you put the mutations in, you introduce other mutations, even though you haven't put the ones in. And so it's very hard. It's been painstakingly slow trying to recreate mutants and work with them. Well, we're trying to actually find out how this diversity takes place. And what we found is really a strange result. The paradigm for quasi-species is based on polio, that it's controlled by the polymerase, the mutation of the polymerase actually changed the virus, so polio loses it, changes quasi-species. So if we do the same with yellow fever, you can take other genes and make one mutation, and you get quasi-species back in different genes. So it's not the polymerase for yellow fever. And I think what's happening is we have a replication complex consisting of 4 different non-structural proteins, and so mutation in one affects the whole complex, and then you lose the proofreading. It's, as I said, it's multigenic attenuation. It's not easy to work with.
Any other question? Like we have a bacteria bank, cell bank, a tissue bank, everything. There is no provision for the viruses for storing them so that
I'm sorry?
for keeping or storing these viruses from parental or primary.
Yes. So the problem was that when the vaccine was first made in 1937, nobody had any ideas about seed lot systems. It was just a free-for-all because there was a real need for a yellow fever vaccine. Hundreds of thousands of people were dying, and so I took the vaccine, get the vaccine there. And then they found after that they had real problems. Serum that was put in the vaccine to stabilize, it had hepatitis B in it. You passed it too much, it became over-attenuated, and that caused the change of the seed lot system. And the primary seeds are held by WHO, as well as the manufacturers, so that when one runs out, they can get more of WHO. Does that answer the question?
Yeah. So, if there is no question, thank you very much.
Okay, thank you.
Very nice.